Now can you hear me? Okay, good. So, first of all, I would like to thank the graduate student that invited me to give this talk. And I will give you a bit background of myself, what I have done until now. So today my presentation will be mostly about crop roots, soybeans and corn in Iowa. So a little bit about my background. So I have my undergrad in plant breeding and genetics from Mexico University, Antonio-Ordonez, that is in the North Mexico. And I got also the opportunity to be involved at CIMMYT Mexico to work in the program of the maize physiology program, actually, doing a little bit about breeding and putting together breeding and physiology at the same time. So I was doing there was selecting genotype for resistance or tolerance to drought or low nitrogen at that time. After that, at the time when I decided to go for grad school, I did my master and PhD in crop physiology. Actually, this is something that probably many people didn't know about that, but I did crop physiology, I did plant breeding at the beginning. So now, here in Iowa State, I'm working in roots. I don't know, I think everybody know, or many people call me in the corridor like Dr. Root. But something that I don't know how that came up, but probably easy. Having a lot of sampling around Iowa, probably that the name came up. So after that, I want also to let you know what I'm thinking of, what is my motivation in my life, in my research career is a little bit of the society, a little bit of farmers, and science. This is what motivates me the most. So every day that I'm doing things, I always think in that way. So a little bit, we have to think about farmers, how to improve yield. We need to think about society, how to provide things that what we are doing in terms of research, that society can also take advantage of that. And finally, the most important for me is science. Whatever we are producing should be published. So this is what made me happy or made me to think about it. So let's go into the topic. So I'm working here in roots. So what is important? Why roots is important? Why we have to study roots? One of the things is because provide the necessary is the connection between soil and plant. So we need to focus on that. And the other important thing about roots is like had a lot of impact in the soil. Why? Because it's one of the sources of carbon, so the carbon pull to the soil. And the other thing is because the attributes such as depth and architecture also is related to the nitrogen leaching. So all these things are connecting all together. So that's why it's important to study roots. And here in this picture, we will see something interesting here. They are very well-looking plants. So this is the point of what breeders are doing, improving yields somehow. And they are focused always, or we are focused, I would say myself also including that, because we always try to select traits that will improve yield, and we focus on that. But something important, we have done a lot of things. We are studying crops, and we figured out many things, but in the, I would say, last 10, 15 years, improvement on yield, we didn't show that much. But so one of the important that now we have to start looking at is the root, because whatever we can see above is good, but in order to be able to improve yield, I believe that we have to look more deeper, and here is. From now on, I believe that we need to put more effort in root systems to understand how the root work in order to be able to produce or to increase yield. This is one important thing. But the other thing, is like, what is the contribution of that root to the soil? So we need to put all together and be able to understand how to do better management practices and how to improve yield. So it's important also, since my talk today is all related to roots, it's important to know that corn and soybean have different root systems. One has a taproot and the other had a fiber root, that they are, at the end of the day, different in their structures and the architectures. So in the case of corn, has the brace root, crown root, seminal roots, and primary roots, and more things. And the soybean has the difference between all of them. That is, taproot has the lateral roots and secondary roots. So this is the most, I would say, the basic thing that we have to know for now about roots. I know that there are many efforts in research people that they are working more deeper how to try to understand any part of the root system. For example, the range root and the length of the root, the angles of each root, and all this kind of thing that they are more molecular on the small scale. So people are putting efforts on that. So in our situation here in Iowa, when I first came here to the university, I found out that because we tried to work with Soterion in relation with Soterios and Mike Castellano and terminal roots, and to try to figure it out and to fix some problems or to improve APSI, in this case for simulations. We started looking for reference and information, and we found that there was nothing there available. So based on that, I had to change a little bit my mind, because I said, okay, we need data, but there is nothing. So we need to work hard on that to try to figure it out how to collect that data. So this is kind of some of the reference that I found it, and the limitation that was like, some of them, there are information that very shallow root that, but that doesn't allow to understand what is going below more than a meter, for example. So based on that, and we start creating questions, and we start looking what is needed here to be able to improve in the root studies or the root research. So one of that was that we want to know what is the root from velocity. What is that? Is the speed of elongation of the plant, or the root, sorry. How will be the speed that root will be able to explore a low or deeper soil profile. And then we found out that there is no easy to translate that data that we are collecting in the field to a unit, like in the soil, per unit area. So we had to understand where will be the best point or the best area to sample it. And then we also found out that it's important to know what it will be the relationship in the water table and root. So there is the relation in the oxygen, like the oxygen. So the distribution, the root distribution across profile, that is also important. And the ratio between root and shoot. And also we had studies, or we want to answer questions that is related to the nitrogen rate, and finally root growth over time, and root decomposition. That we saw that this is the thing that we had to know, that we need to explore deeper to understand this kind of thing, because since here in Iowa, what is important as the state is the production, the crop production. So we had a lot of things to improve, to keep going and do better. So this internal root, that is the question that it came to my mind at that time. So here is a map that will give us a little bit the idea how will be the distribution of roots. Every time that when we talk with people, and they say, oh, in the west side, the Iowa, I expect that root will be deeper. Why? Because the water table in Chori is deeper. And as you move to the west, to the east, sorry, it will be shallow. because the water table is shallow there. So this is what people are commenting every time when I had the opportunity to talk with them. I always had this idea from them. So this is my hypothesis, and basis on that, basis in the water table. When we had the shallow water table, the root will be shallow, or the root system will be distributed in the 60 centimeter, I would say. So with deeper water table, so the root probably will follow that pattern of the water table. So this is important. And since our research was focused always or mostly in the root system and the interaction with environments and mostly the water table, we saw that it was important also to get that information. So what happened here, so we put it in the soil in every place that we were sampling, a water table to measure the dynamics of the water over time. So about root depth, we developed an strategy how to be able to measure how the speed of the elongation will be in that plant. So we were using a simple pro that many of you, I'm sure you know. As the soil science guy, you are using a lot of that. So we were collecting every 2 weeks in some places and every week in another place, depend the distance, because to travel across Iowa is a bit difficult. You represent a little more money to be able to travel here or there. So we were doing every 2 weeks in that case. So internal root mass, we were using this approach to collect soil core in the field, and we were again traveling everywhere in Iowa. In the previous maps, you were looking in the stars that indicate where we were collecting that root. So, but it's not just like that. We collect root, and the second step I would say is to wash that root, to clean, to be able to get that root from the soil. So that is the system that we had, and that in collaboration with also with the USDA laboratory, that they had that instrument or that device, I would say, that we were able to use and to be able to collect that root. So I would say that this is my protocol that I was using to be able to get the final root. And here we see Dr. Root preparing the samples that will be analyzed to be able to get the root length and the root mass, because that is the most important in our studies, to be able to get that information. Oh, so important, something good. So the final step of our process or the protocol will be the carbon and nitrogen analysis. That Emily is in charge for that aspect on our research. So all this is our protocol that we use to be able to get all the information. So from now on, getting into the science part. So this is all the question that I had at the beginning. So I tried to answer all of them. So this check mark will show you what the state we are. And some point will appear some little box there that will indicate also what we are doing and what state we are. So the first question or the first problem that I found, it was that the root depth. So we focus on that at the very beginning to be able to understand where the root will be during the season, how fast will grow, all these things. So here, this is one of the slides that I like a lot, because we were able to understand or to put together aspects that happen below ground and above ground, how the root will grow, and also what is going on above ground. Different stages of the plant, so that had, at the end of the day, some connection with each other. So here we can see what the root will stop growing or will stop exploring more soil when it will reach the water table, and also occur when start the grain formation, I will say, or the yield formation in the crops. This happened both in corn and soybeans. Then, all the data that we collect for corn and soybeans in this graph is summarizing basis that we can see, importantly, that the root elongation had 2 or 3 phases, 2 important phases, I would say. One is the phase of the slow and the faster one. So the same happened for both for corn and soybeans. And here we can see that both crops, they had more or less the same depth. They explored soil almost at the same, I would say, soil profiles. And happened for both of them. So here this graph, this figure summarize very well what is happening and when occur all the changes in the field. What we think, for example, here we see that the roots change the elongation rate when we had, this is leaf number and node numbers in corn and soybeans. So the rate of elongation occur more or less in that part that will be the number of leaf and node. And will start growing at that stage. Will be around 18, that is more or less for corn and soybean, for corn R2, and for soybean will be R5. Is the time when the root elongation finished for both crops. And the important part here was that we also find correlation with the water table. So the maximum root depth in both crops are dictated in part by the water table. So when reach the water table, the root cannot go down because there is lack of oxygen mostly. So going to the next problem, I would say that at that time we had was how we will translate that information or that root that we are collecting in the field into real value that we'll be able to use. So for that we developed one algorithm that will help you to be able to delete error samplings, and that will help you to choose the proper points where you can do your sampling, and the values that you will get will be representative of this area. So for us, this is the problem. I went to the field, we organized with the students, we went to the field, we collect soil cord, one in the row, and the other between a row, 2 rows. So if we found that the root mass are different, so they are not the same. So there are variation from.
When did you do this? You just showed us the time that it took for the roots to meet between the 2 rows. When did you take these samples?
That samples I collect at R2 for corn and R5 for soybeans. So this is the stage that we get it. So the important here is like both points are different. We get different values. So the question here is which point you will select to be able to get the representative values. Anybody of you know that? Where you suggest to go and sample? Where you will send your students say, hey, do there, that point I want. Any of you have an idea? Halfway. Any other idea? No? Okay, so we'll find here. So one of the things I found, because I was like excited, I want to start working in apps, input data in apps and start doing simulation. But and then I say, no, how is possible to do simulation if we don't have one value that will represent the area there? So I start looking in the literature, surprised, we find the same thing. So in the literature, they sampling in different points. There are no uniformity, like people are sampling wherever, I would say, to try to get some value. So there is no uniformity. So after that, here is the answer. So we build our experiment and we collect soil cores from different positions, 0, 12, 34, and 36 centimeters. So, and then after that, this is the representation how we got all the measured mass in length. So everything changed across the spaces. So this is the values that we get. So to be able to eliminate that errors, we found a model that will be able to simulate that point, and was one exponential decay model that I used, and to fit in that one and all the points, and we found this is simulating points. I did a simulation every 2 centimeters and fit in that way. So that model or algorithm that we developed allowed us to calculate the weighted average. So that will be like when I would say the proper or the maximum or the accurate data that we can get from that sampling. So important here is like having that model, when we fit, we were able to understand how much will be the difference above and under or overestimation for our sampling. So the sampling in row, that is these ones, always overestimate our root data mass, and between the two rows, underestimate. So you guys tell me which value you will use now. So don't worry, we have a solution. So my model showed that the best point to sampling, it will be about 10 centimeters distant from the row. And the most importantly that we provide here was this simple model. So for now on, I encourage you to be able to use that model, that you will just need to use the value that you collect in the field, and then use also some index that we calculate, and you will get your proper values. So the error will be eliminated from your sample using that algorithm. But important, remember that will be always around R2 and R5 for both crop. So this is what we provide. So from now on, we are confident that our data that we are collecting will be okay, at least the information that we will get from there. So we eliminate errors. So the next point will be, so here we see the little boy showing in what state we are. So this is a paper that we are working now, and Dr. Elnas is working and that manuscript with collaboration with Sotil and all the other people involved on that. So that was one of the important things that we want to do it, because when we simulate in absence without fixing or without calibrating some stuff, there are some algorithms inside absence, we will see that absence simulate here without stopping. So we need to find an index or how to be able to let absence know that how to stop when we'll reach the water table. So the blue points here is measure the water table and simulate the water table. So with that points that we collect in the field, we were able to let APSI know that when had to understand that there is a water table, it had to shut off the elongation of the root. So this is one of the important aspects that we are doing, because our data that we are collecting is always to try to improve models, crop models, in this case absence. So going to the second, or one of the important part, it will be the root distribution. So as you see, we are collaborating always with different students, grad students and postdoc students. We had that opportunity in the group. I will say my small program, I will call it that. We are working that way in collaboration with everybody. So now I would say, based on that map is the real information that show how is the distribution, the water table across Iowa. So as I mentioned before in the previous slide, that this part, it will be the deeper water table, and that area, the blue, the dark blue, will be the shallow water table. So everybody expects that the root will be always shallow in that area, but our research show different things. Let's go there. Let's go into the result of the information that we got over there. So we collect information or data from 19 core data set and also 12 soybeans across Iowa. So all that stuff, there are the sites that we collect all this information. And the orange color represents the area that we had water table. So this is the important part, I would say, that we provide to the community. So we have here some values information that in the literature that if we review a little bit, we will find different values or many things. But the most important is like how deeper they want, from where that value is coming from. So here we have the opportunity to present or to offer to you that how will be the distribution across soil profile and how deep that will be. This is our values that we got from there. The root length, for example, it will be in that range from more or less 100 to 200. And CN ratio is kind of the same thing, but is uniform across the soil profile, doesn't change. But important when we compare corn and soybeans, there are differences. So going more in depth of the thing, or I would say the most important information, I think, for the soil science guide, that would be a very valued information. How is distributed that root mass in the soil profiles? So we found that this value, it will be around 86% and 81% in the first 6 centimeter soil layer. So I think that data is more or less familiar for many of you, for people that are sampling, doing sampling. So all the distribution is in that way. So maybe that value is something that for you will be useful if you are planning in the future to collect root, or if you are planning in the future to write something. More or less here you can get some idea how the root distribution will be. So the important here is not just how the root is distributed. The important is to see also how it, I would say, how will explain the weather condition, this variability in the root distribution. So here we had that the water table play important role in the distribution of the root. For example, that was 30 centimeters. So there were correlation between water table and the root mass. But the important here is like the effect is different for both crops. They are not the same. So I think that happened because they have different structure or different root systems. So both respond different to the environment. So this is, I think, very important information or very important knowledge that we provide, like to understand how plant will behave depending in the root systems, are different or not. So that is probably one of the important information that we are providing here. The next, we have the next. So that will be especially root shoot ratio. Why special? Because root shoot ratio is what provides the information how your plant will do in the field, above and below ground. So probably this is one of the important information that I had ever seen in the literature. Why? Because we had opportunity to measure below ground, and also what is above ground, storage, and final yield. So in the literature, I'm pretty sure you will not be able to find this kind of information. And most, or what is important that is because this was collected across Iowa from many places. So all this will give us the idea how will change all that parameters or variables across Iowa. So they are not the same. So this is important. Roots will be always in about will change from 1.5 to 2 megagram per hectare. And the yield will be about 10 to 15 megagram per hectare. And this is our values. Probably this is not very familiar with most of you. So because there are not many information in the literature that will be able to measure that things. I think here in Iowa State we are pioneered because we are collecting that information in the field. Most data that we can find in the literature is basically an experiment that was done in greenhouse or under controlled conditions. So here in agronomy department we have that thing that we collect in the field. So I think that is the most important thing, that we provide science, we provide information on how people will be able to develop or to continue doing science. So this is the most valuable aspect that in our research. Or the
Was Muscatine irrigated?
good point. Yeah, I forgot to mention that. Muscatine is irrigated. So in all our experiment, we try to cover all the growing condition that we had in Iowa. Tile drain, no tiles, tillage, no tillage, and irrigations. So that all information is in that graph. So all this variability that we had is made it, or is coming from that, from the environment. So the same happened for soybeans. The same thing we had for soybeans. So we didn't work just for corn, we had soybeans. So wherever do you want or do you need to know about corn and soybeans in terms of root system, we had here. We can contact Soterios or Mike Castellano, and we'll be able to work with this data and to continue doing improvements and any crop model that we had. So keep in mind that all these parameters will change based in the growing conditions. So they are not the same. Don't use in your model that 15% will be the root shoot ratio. It's like happened for harvest index, and many researchers that say, okay, just pick one value, what, 50%. So here we had the real value. So this is had variability across the environment. So, and this is our value that we found. So here, I think when we compare both crops, we can see the difference, how they change each other. So root mass and root length and all the parameters that we measure, they are mostly different in both crops, but except the root nitrogen uptake. Why that happen? Simple, because corn produces more root compared with soybeans, and the nitrogen that the percent of nitrogen or the nitrogen contained is higher in soybeans. So that's why they are at the end of the day collecting the same amount of nitrogen from the soil. So this is our information that we are providing. I think this is what you guys have to know in terms of root system. The difference between corn and soybeans. And it was not just like that, we also tried to figure out what variable will explain better the variation in root mass. So we did correlations between root drive weight versus all these measured variables. And we found that mostly they are correlated with variables that in some point are dependent on the root system. But it's important also here to see the nitrogen uptake that also correlated with the root mass. That explain or that give us a lot of good information. I believe that this is something that we can use or we can be able to use in the future. But interesting here in the graph, I don't know if you guys noticed that soybeans had some correlation with the environment, but no corn. So I don't know how to explain that, but it's something that I think we had to look at it more in detail and to see what's going on there. So here we have that correlations that I believe it is something that we can continue exploring more deeply and to be able to get good explanation. So the other part that we had is everything related to nitrogen rate, because nitrogen, as we know, is one of the important nutrients that many people are using in the agriculture. So here we run experiment across 4 environments or 4 sites. Here better to explain in that map. So we use 4 sites in McNair, Ames, Kanawha, and Muscatine. Again, Muscatine because we had irrigation system. And the other because cover more or less what is the gradient of change of the environment across Iowa. So here we explain how is the percentage of sand for each location. So an extra information that we had there in Muscatine is that we had sandy soils. So we want to compare all these changes or all these factors in terms of nitrogen rate. There we go. So in that figure, do you guys see something different or something that you were expecting? I don't think so. Everything makes sense. You increase nitrogen, you increase yield. More nitrogen, more yield. So what happened here with the root? More nitrogen, more root. More nitrogen, we lost root, driveway. So it's something that we discovered, I would say, in the root system. At the very beginning, when I started analyzing that data, I was working with, I think it's the red one, Ames, is the first experiment that I started cleaning. When I saw the result, I say, when I measured the root mass, I said, something wrong here. I was expecting that the root mass will increase, the response will be the same as above ground. I said, no, I think I did something wrong here. I don't know what's going on. And then I had the opportunity to get the second experiment, and I saw the same result. To say, oh, something is wrong or is the reality. And the first was the same thing, we get the same trend. The more nitrogen, the biomass decrease. So it's something like, I can't really know what that may happen, but this is something that we need to explore deeply. Why change the root mass? Probably that we need to look at in the terms of physiology. Probably that will play a really good role to understand this kind of things. And here I put only that graph because in the previous one we were putting only 1 year, but at that one include 3 sites and 2 years. So this is like reinforce what was our finding. The root will always decrease when we overuse nitrogen. So I would say that there is no need to throw more nitrogen that we need, because plant will not be able to get it, will lose to the ground, and later probably will have impact in the environment for the excess of nitrogen. So this is what we got from that 2-year experiment with 3 locations. So everything was consistent. So I didn't expect something different for the nitrogen uptakes. The more you increase the rate of nitrogen, the higher will be the nitrogen that you will recover from your crop. So that, I think, is made a lot of sense with the reality. So I will say that my presentation is done. I tried to do it short because I didn't want to keep you for long here. I tried to explain a little bit what I'm doing. I think the other 2 parts of that, I will keep it for the next opportunity. If I have opportunity to present again here, I will keep it, and I will explain what is going there. Just I will give you a small introduction. Probably for the next one is like, it will be better to make a question to you. How long do you guys think that will stay the root in the soil?
Which root?
That's a really good question. The core root or soybean root, how long will it stay in the soil, or the rate of the composition, how long will be? Do you have an idea?
2 weeks.
2 weeks?
Yes.
Pretty close.
What we saw in the residue, to have an idea of what is going on, the residue probably takes at least almost 3 years to totally recover. But in the roots, it's a different environment. I don't know. I believe that maybe it takes a similar time.
Similar time. Oh, that's a really good point. Emily?
Corn will last longer than soybean. No time.
Okay. So I will give you the answer for the next presentation, but I will let you know that I'm collecting also that information. We clean, I will say, in the field an area that from the previous year that I'm collecting roots. But I will let you, or I will want to tell you that I didn't saw any change from the previous year until now. So the roots are still there in the soil. There is not very big, or the rate of the composition is not that fast as I was expecting when I designed that experiment. So I found roots. I forgot to bring the bottle, but I had in the lab roots from the previous year, and they look happy. I know that they are there, but they are well looking still. So this, I think, that will be something that we need to explore deeply and to understand how will be the rate of the composition, because I believe that had impact also for the carbon budget. How will be the rate of the composition of the root, which is part of, root is one of the, I would say, part of the carbon, organic carbon in the soil. So I feel that this is something that we need to explore also very deeply. So what we learned today was simple. Root had 2 phases of elongation, 7 centimeter per day, 0.7 centimeter per day, and in the fast rate will be about 3.2 centimeter per day. So this is the speed of the elongation that the root system will be. So corn and soybeans kind of the same, but soybean had a little faster rate in the first phase. It's only the difference. And then the maximum root depth is strongly determined by the water table. And it can grow in soil, in saturated salt. So at some point, the root will sense that it will not be able to continue exploring more soil, so it will shut off the elongation. So, and then we have the optimal sampling point that will be 10 centimeter. And then the other important thing that the root core in soybean had in the first 60 centimeter, about 80% of the root are distributed there. And also the root shoot ratio are not the same across the environment. That change is affected by the environment. I think this is a lot of room for improvement from the plant breeding point of view. So if we have variability, we have chance to do selections, or we have things to work for. And then corn has higher root dry weight compared to soybean, but lower nitrogen. And the final, I would say, that we learned was that nitrogen rate affects root mass different than yields. That, I think, is pretty important thing that we had to know, or something that we found. And what is next? The next is, for me at least, I would say, because actually this is like kind of personal information that I had to give it to you. So one of the things, I had nice size. I want to know what happened in 99 counties. Is that possible? That the values change or are more or less the same? And why not to extrapolate from 3 years to 33 years? That what is the value of what we need, or the number of years that we need in order to be able to do modeling when we expect that will be changes in the environment. This is the required numbers. So this is what I wanted to share with you today. I hope you guys got some knowledge from here, and I will be happy to continue working in roots, and hopefully we'll be able to present here the next year. Or I can say that if I got an invitation from wherever I will be happy to come back and give another talk. So I want to finalize my presentation with knowledge my collaborators and the 2 labs that, actually I forgot to say that I'm working for 2 labs. So I need to keep happy both group. This is not that easy. Anyway, so this is all the group that I'm working with, and I think we are having a lot of fun collecting root, and we try to pull all the information together and get out papers. That is the most important, I would say, for my stake. So thank you very much for your attention. If you have questions, probably I will be able to answer. If not, we have Sotirios or Mike Castellano that can support that. Thanks. Yes.
A nice job. We are rooting for you. How was you got on one of your slides, your series of slides, you were showing root growth as a function of an application. And I noticed you had one segment that said in row. Nitrogen as well as phosphorus applied in
Which one was that?
Further forward, towards the end of the slide, we were talking about the nitrogen rate impact on root growth. That's right. There, you have one thing wrong.
In row.
Well, how were the other ones applied? Nitrogen, especially nitrogen and phosphorus. The reason I'm asking is the concentrated areas of the nitrate ion or phosphate ion will cause root proliferation in that specific area. And if you get proliferation in that area, it reduces growth in other areas of the root zone. I'm just wondering if that fertilizer has been banded or if it was uniformly distributed.
Yeah, and We had, I would say, different type of fertilizer. I think so. We were using broadcast nitrogen on that experiment. So I will expect that the root, as you said, will will grow, as you said, where the nitrogen will be most accumulated. But in that case, it's not possible because the nitrogen was distributed uniform in the area in some of our places that we were working with.
Did you look at above ground shoot mass in this experiment? You looked at yields, are you also increasing? In other words, I'm wondering how harvest index changed nitrogen.
Yeah, I didn't have done yet that exercise. I had the information about yield, and I expect that it will be kind of, will not change that much the ratios. Yeah, will be in terms of yield, for sure. They will decrease the harvest index basis in the nitrogen level. But we don't know yet with uncertainty what is going to happen. It will change the ratio between root and the final yields. Yep.
Raziel, the slide where you had the root to shoot ratios for corn and soybean, can you go to the
That one?
Yeah, so the slide here, it seems to me like the harvest indices you have here for all the soybean, there are a lot of other things. So you have the root to shoot ratio, but what's the harvest index range?
Yeah, the harvest index here ranges from, as I remember well, about 45 to 55% in some places. So it's kind of good range, but that is more or less determined by the environment.
Right. So the corn data look to be around there, but the soybean data look to be much less than 0.5. Maybe I'm doing the interpretation correctly. Is that right?
Just Raziel, with this equation, what is stover biomass over there? Is the maximum biomass or is it maturity?
So this is the maximum biomass.
Okay.
Maximum biomass.
So in terms of maximum biomass, it's much less than 0.5, but at harvest it probably is value.
It's kind of the same.
That was my question.
So any other question? Oh,
Your root to shoot ratios, we've had these conversations before, seem to be pretty low compared to what has been published in the past. Does that suggest something about the genetics? When the genetics is targeting increased productivity, in other words, root systems may be the same for today's hybrids in terms of mass as we had 20 or 30 years ago, but the above ground growth has changed.
so I will say that there have been a lot of improvements in the breeding programs, and they are targeting to get less root biomass and put all that together. So it's kind of to try to make a root system more functional, because we know that when plant is putting more biomass below ground, it means like energy loss. So I think there is something related to what you said. So the improvement of the plant breeding to that things to make functional more to the compared with the past. Now I believe that root had a lot of changes, like becoming more thinner than in the past.
So something I want to add about the nutrition ratio. We should be very careful when we read values from any paper or presentations. This is a value that is time dependent. Raziel measured the roots when we had the maximum biomass, not the composition in the roots, nothing. Usually when they make a publication or the previous studies, they report the root mass usually at maturity, and when they collect the shoot, usually they don't have all the leaves. So we need to be careful with this. I don't disagree with the genetics. Something has changed there, but I don't have data to support.
I think to put that into perspective, Rick, think about the the maximum amount of stover that is ever harvested from a field, and compare that to the harvest index, and it's just, where does it all go? We don't know. I'd love to solve that question, please. Kind of a piece of this.
I just wanted to comment on what you talked about with breeding, because this is something I've been thinking about as well. Is in our breeding programs, we select for plants that produce high yielding, and we feed them appropriately or to whatever the production practice at the time would delegate. So if growers are going to be putting on 200 pounds of nitrogen or whatever they are, that's the selection criteria that we're putting on our process. So coming from the breeding side of things, we are breeding for smaller roots, as you can see over time, because we're babying the roots, we're putting the nitrogen where the crop needs it, and even now with more precision farming techniques, we can put the nitrogen right where the crop needs it when it needs it, and therefore the roots don't have to scavenge. Similar to your 300-pound nitrogen application, the roots are smaller because they're not having to go and scavenge for the nitrogen. It's all supplied right there for them. So our breeding practices are doing the same thing essentially. That's my thoughts on it, at least.
Yeah, that's a really valuable point. And I will also add to that comment that when you have enough nutrients in the soil, I would say that the plant will not need to produce a lot of thick roots. So mostly will produce the masses of root in that area that you have the most nitrogen, but it will be thinner, so more active. So that will be able to get all the nutrients and to pull all up. So that is one of the things what I'm thinking, that plant, they don't need to produce a lot of roots, because that we know. So, from the perspective of breeders, we want plant that produce lower root, that invest less energy there, and to be able to capture as much nitrogen they can, or any other element that is necessary for the plant. Yeah, so actually I'm fascinated with that paper, that deep in seed, but because they provide quite a really good thing how the roots should be. That is something that really interesting, how we believe it, that root has to be. But I will add to that point is like, how should be the soil that you want to grow that root or that plant. So that will change. So we have hypothetically one kind of root that we want, but we don't know the environment. So it's important to work also in that aspect, to understand where you will plant your.
There's a lot of environmental interaction, as you can see
Yeah.
in this chart right here.
Yeah, so that's really important information. I think from the plant breed perspective, you have a variability, something that you can continue working and to do breeding for. If we look at that way. So now the important is to understand how much root will be in that soil, or how much root do you need to optimize your yield or maximize.
Any other questions? Otherwise, I think we'll wrap up. Thanks again.